Well, everyone, first off, Happy New Year, and welcome to the kickoff presentation for the 2025 Spring Science Circle presentation series. I am your presenter, Stephen Gasior, also Zootfly in Second Life. One talk that I always give at the beginning of the year has been a tradition for about 4 or 5 years now of what was interesting and what was happening in the area of genome editing for the previous year. I picked this topic because, one, it helped me learn the topic as I became a working professional within the field, which started also about 8 years ago. And then it's just a great thing that has so much relevance to think about technology, people's lives, between therapies, between agriculture, and just a lot of interesting stuff that's really happening. And it's something that the invention of this technology was just published in 2012, or at least the main CRISPR version. But as you might notice from the name change, I used to call this the CRISPR year in review. And so now I just call it genome editing to have a more expansive topic because it's more than CRISPR these days. And so with that said, the overall structure of this, what I think are the good things to cover, again, just a quick reminder on the basic nuts and bolts of what genome editing is, how it works, this new CRISPR version that has made it so much more popular and easy to use. And then I usually focus on therapies or things that are coming to the consumer. I talk about how these materials are getting into cells and how scientists are developing these to get them into and be effective. And then target genes, modified organisms, these are the ones like, oh, what did somebody do? What was interesting? Why are they modifying this? Or what's the impact? Again, these aren't necessarily therapies or things in the field, although in some cases they might be. Now, it's a rapidly advancing field. So I always talk about new tools, and I have to admit this is the section, I put it near the end. You might feel a little bit lost in some of these. The key thing from new tools to take away is just there's a lot going on, and just try and take away the key impressions that I'll give. And then finally some cautionary tales, because this technology being very new does have some issues that still need to be ironed out. So one other thing I'll say just in general is I'm trying to cover as many topics as I can, as kind of briefly, but as clear as possible. So, the slides will be available. I'm happy to talk about it other times, but just don't try and get too bogged down in trying to learn and know and remember everything. It's a lot of it's about the impression of what's happening and what's interesting. And then the other part too is the way I structure these. I have the title of the papers usually, or the titles of the press release, and that usually is highly informative. And then in white, I will be quoting something usually from the paper. And then the green text will be usually what is a quick one-liner takeaway, too long didn't read version of what you need to take away from the information. So with that, this will hopefully be fun. I do have an opening statement as well, which is, again, I am a scientist, a molecular biologist at a company called Corteva Agriscience. And we do operate in the space, and we have significant resource investments, product pipeline, developments are ongoing, a lot of intellectual property representation in the space. I'm not here, though, to be a representative of the company. I'm not making statements about what the company's positions are, and nothing here should be considered like investment advice or forward-looking statements. All right, so a little bit of background on CRISPR. And again, I'm making the assumption that you have no background in this, and I hope that slide is resolving for everyone. It's not resolving for me. That the development of genome editing was dramatically advanced by the discovery of something called CRISPR. And this was awarded the Nobel Prize to Jennifer Doudna and and Charpentier for making this a very accessible technology. And the main idea, the main player you'll have heard about is a protein called Cas9. And so what is interesting about Cas9, it's the one that's most well known. It's the one that you'll hear about. There's a lot of patent positions on it. But what it does, it's able to edit DNA. And then within this broader category of genome editors, there's another category of these known as Cas12s. And these are distinct from the Cas9s. And I'll talk a little bit about that in a second. But these operate the same ways, that you can program them with RNA to interact with DNA. And then there's a third category, broad category I want to mention right now, which is Cas13. And what Cas13 is able to do is actually edit RNA. And so RNA editing is not as practical in many cases in terms of products and therapies and whatnot going out in the field, but it will get some mention as well. But these all share the same general feature that you can tell them where to go on the nucleotide of interest based on sequence. And then they do stuff. Now, one of the main things that they do is that they interact with DNA and make a double-strand break. And that double-strand break can lead to consequences in the cell. Now, these consequences are dependent on how the cell reacts to the double-strand break. And again, we'll talk at the very end about how that usually has many implications for understanding the cell's reactions to these and where there can be deleterious effects. But in general, when something happens with that double-strand break, it can usually lead to a targeted change in DNA that we're trying to accomplish something. Now, I have a couple other things that are shown on this slide too, which are ways that these proteins go to DNA. And then instead of making this potentially very dangerous double-strand break, they'll do things like make an edit. So they're just making very small and minor changes to the target sequence. In this next slide, what I am presenting here is a reminder that genome editing didn't start with CRISPR as a technology. What existed before were things known as meganucleases, talons, and zinc fingers. And these were based on other ways that endonucleases could interact with DNA, and they were programmable. The thing is, they weren't you couldn't just give them a template. They actually had to be engineered to recognize the sequence that you wanted it to go to. However, there are advantages to that, and I'll talk about that later, that in some cases, the fact that you don't need the RNA means that it's easier to deliver. And then there's and then what I want to just focus on a little bit here at the bottom is that there are the derivation of the Cas12 and the Cas9 proteins that are our main engineering tools. These are actually derived from older versions of these proteins from bacteria that are known as TNPB and ISCB, and these are things related to transposons, or things known as mobile DNA. And these have been evolved in a way to do what they do now. But in fact, one of the things that researchers are doing now is going back and asking, can we find more examples of how well these work from other examples in genomes and in sequences that we're discovering. So again, just one thing to keep in mind is that there's a broad category of ways to engineer proteins that are out there, and that we're co-opting them from their natural behaviors in order to become genome editing tools. All right, and here's a reminder on how they work, which is you have your protein, and what you provide it is typically an RNA sequence that is shown here in black. And that RNA sequence, along with the protein, can go recognize and bind to its complementary DNA sequence, and that's what's red. And so that's how you get this very specific interaction of where the protein goes and recognizes a specific sequence. And then usually, again, in the native cases, it causes cleavage, it chews up the DNA in some way, and then there are consequences to how that gets interacted. Now, in terms of CRISPR and Cas12, bacteria generally use these as a type of very primitive immune system. So what they're doing is recognizing DNA or RNA viruses that are coming into and trying to destroy the bacteria and using that as a way to destroy the invader. And that's why it has the sequence specificity and the ability to learn what it's interacted with. And I do want to point out again, so this is the diagram that also has, when you have this pairing and you're telling the protein to go someplace, you can do stuff other than make a break. And that includes something known as base editing. So again, if you imagine you've made a typo when you're in a word processor, think of base editing as a way to just go back and substitute in the right word, just correct that one letter. And then prime editing is an example of something where, imagine again, you're writing a document and you realize you accidentally left out a word. Well, then prime editing is a way to think of going back into that DNA and just adding a short snippet of information, a short snippet of letters, and adding that back in. And so these are very powerful tools in order to make changes to cellular, to genomes. And this slide is completely blurry to me, so I will read it from my template. Let's talk therapy advances. So this is always, I always like to lead off with this because this are the ones where people ask, how does this potentially relate to me? And so the first one where something kind of interesting happened in 2024 that's kind of unique and novel compared to all the therapy stuff that had been happening before is a therapy known as NexZ, in which patients get injected with a Cas9-based product. And what it's doing is it's targeting a bad version of the gene. And so what this is doing is keeping an aberrantly forming protein known as transerythrin from forming and aggregating and causing problems within a person's blood. And so by disabling the ability of this thing to get expressed and be made, it does a really nice job of allowing and alleviating the symptoms. And what's unique here in 2024 is this demonstration that a single infusion allowed for a persistent response. And on top of just marking a reduction in the serum levels, they're also reporting in this clinical trial that the disease markers like cardiac arrest or other heart situations are actually also being alleviated. And so again, actual clinical effectiveness for this particular therapy. The other one was for retinal degeneration. And this is, again, some of the low-hanging early fruit for CRISPR gene therapies is trying to disable a bad copy of something. And so this version of retinal degeneration is something where a pathogenic version, a bad version of the gene that makes a protein doesn't allow photoreceptors to align and get nutrients. However, and so this causes early childhood blindness. Now, it turns out that these patients actually still have a decent amount of functional receptors So as long as even an adult, you can remove this misfolded protein, they can recover some degree of vision. And so that's what this editing therapy was done as well too. And so again, this is another example where, uh, again, the clinical trial was just to show, is it safe? Does it has some degree of effectiveness? But this one actually showed, and they measured a correction of visual acuity, where again, these patients are getting nice function back. Again, another thing that's kind of interesting about the eyeball as a clinical intervention, and this is a funny aspect of these trials, is they always treat one of the two eyes, and that way, in every single clinical trial, your control, your negative control, is that exact same patient's other eye. And so, one thing that's nice about these is you can generate usually very reliable, robust clinical data, even though you might be saying you're only half helping the patients. But again, this is a good example of something where, again, showing good alleviation of the symptoms. And then the other one is an interesting one where, again, they're trying to knock down the expression, in this case, of a normal gene. But the normal gene they're knocking down is something that processes a bad working of a gene. And so that way it doesn't get involved and accumulate into a state that gets impactful. And so the idea here is that hereditary angioedema is due to this buildup of bradykinin. And there's this, what they're doing is knocking down a protein called kallikrein so that bradykinin doesn't become pathological. And again, the highlight of this one is that a single dose was able to create a long-term sustained effect. And so again, a lot of the text here is just describing that, again, what the disease was, but they actually are targeting the idea that a single dose is giving long-term, lifelong control. How bad is my voice for people? Because I don't know that I have any control over that. I'm using a headset. Okay. All right. Just double checking. Thanks. Thanks for letting me know, though. And what I'll do is maybe try and not emphasize too much. All right. So the last slide I want to show on this one is just a quick roundup from Innovative Genomics Institute. They're something that, they're a group that usually does science reviews and literatures and can actually give some forward-looking documents. But what they're doing is giving a kind of quick summary of as you go through the different phases of clinical trials. And so, one thing that is notable here is that there is one approved treatment. There are patients who are able to get a CRISPR therapy for sickle cell anemia, as well as some thalassemias, which are, blood hemoglobin type diseases. And so, that's very exciting. Although I was actually just reading a Wired article about it, that there's not been a lot of patients who received it yet, because it's still quite a bit of work to get that therapy. All right, so let's get into the next section, talking about delivery advances. And this is, again, the therapies are great, but you have to realize that it's really this R&D work that's going into not only how well do these function, but how do we get them where they need to be, especially in patients. And so the first one I want to talk about here, now again, I've talked about this a lot in the past, and one of the big challenges for humans is being able to basically inject someone so that the CRISPR payloads get to the right cells, or in some cases it can go to all the cells, and then has its effect and is delivered. And so I've talked about this before. There are viral vectors, or these things known as nanoparticles, viral nanoparticles. And a lot of that's advancing, but there was very little, I would say, dramatically new in 2024, with the exception of this one. And what's unique about this one, so this is a technology where they make small little like subcellular packets that include all the materials you need in order to inject them into a patient. But what's unique about these is that they're designed in a way to not only deliver the CRISPR payload, but you can also package other accessory factors that help out with the therapy you're trying to do. So one example that they give here is that when you're trying to make double strand breaks and insert a gene into that genome, you want to not have something known as non-homologous end joining, the NHEJ, get in the way and do it the wrong way. And so one thing that you try and do is also co-deliver things that block NHEJ so that your gene can be inserted by what's known as homologous recombination. And so this technology is something that helps enable and allow you to co-package other factors into your therapy. So I thought that was kind of a unique and new one. Let's see. Here's another one where there's actually several things kind of interesting about this technology, which is how many people here have heard about and know about the microbiome and how the microbiome in our gastrointestinal tract is really important for health? Yeah, right? It's one of the big news items these days. And so what we would really love to do with CRISPR is find a way to actually genetically target the bacteria in the gut in order to, again, could be pathogenic bacteria we want to get rid of, or maybe we want to enhance the healthy ones in some way or another. And so the this group, and what I have here is a slide showing that they're using something called bacteriophages, and they found a way to package and find the right bacteriophage to be able to broadly target more bacteria. And then that's what that little capsule looks like. It looks like a little bit of an alien or something with legs on top of the cell surface. And you can actually deliver base editing payloads. And what they demonstrated is that they were able to do this with multiple different target bacteria in mice, and able to show by then re-extracting those bacteria that they're actually targeting the base edits they want. That's what the graphs here are showing, where the yellow and blue, where you see them tail down as you get farther to the right on the right-hand part of that slide, that's demonstrating that they were actually able to target those genes when they're adding the payloads. So I think this is something that I think will end up being a really interesting development, partly because you don't if there are places where you can help people health with the GI tract, the amount of safety and regulatory hurdles for developing a pill and not worrying about messing up the person's genome and health effects on the person becomes a much easier hurdle to get through. And so Elaine is asking, how does RNA show Cas9 where to go? And that comes down to the DNA ladder, the double-stranded structure of DNA, and that there's a complement, there's a mirror image of one set of sequences, and if you can deliver that mirror image, it allows the protein to go right to that spot. And so again, think of it as, it's like a photograph, right? In a sense that if you have a photograph of somebody, you can recognize them, and you're going to recognize them really whether it's the mirror image or the flip side image, but it's a way to match things together. So another delivery advance, this time in the world of plants and agriculture, is what are called RNA viruses. And so RNA viruses tend to be, very fragile, but they are quite robust in the plant environments. But one of the downsides, and this is true of all viral vectors that we use, is that they have a very limited size. There's a very limited package you can put into them and have them still work as the virus that's going around to cells and getting into cells. And what I really wanted to highlight in this paper is that there's a version of Cas12. Again, this is not true for Cas9. This is only true for a particular subclass of Cas12 known as the Cas12Fs, as well as I think L. They are really, really small. In terms of their protein size, they're really, really small. So they actually can fit into these smaller viral vectors. And what they showed in this one is that they are capable of delivering this RNA virus. And if you look at the leaves, that if you compare what you see on the left, where they're trying to use and trying to fit in the Cas9 into, as best they can, into this vector. It's not stable, it doesn't deliver, and doesn't genome edit very well. But if you look at the leaves on the right, you see a lot more yellowing, you see a lot more of the loss of green coloration. That is an indication that the editing is working much more efficiently. And so this is, again, looking for there are versions of Cas proteins that can be extremely useful in specialized situations. And in this case, it's the small size helps deliver it. Okay, so those I thought were the kind of main delivery advances in the field. There's a lot going on in that space, but those were the ones I thought were kind of most notable and new. In terms of target genes and modified organisms, so uh, there's a category of diseases and pathologies known as triplicate repeat expansions. And what's happening is within the cells within an individual, uh, the number of DNA letters in the gene are just getting bigger. And then that means, in many cases, that means the gene doesn't work as well, doesn't express as well, or something's happening to it. And so Huntington's disease is a classic example of this. And what this group showed, and that's what the diagram is showing up there on the top, is that there's this part of the gene where the sequence CAG is multiplied multiple times in a row. And what this group showed is that if you target Cas9 to make breaks within those repeats, you can actually get those repeats to shrink. And that, again, the response of the cell, however that DNA repair mechanism is working, it actually shrinks them back down. And that's what's shown in that letter G figure, is that the green bars is showing how many repeats there are. And so you go from a large number, and now you have a complement of versions of the gene where the total number of repeats is much smaller. So that's where you see the green bars on the left. And so, this is telling us a little bit about actually how these repeat expansions develop in the first place. But it's also a very nice example of you can have a pretty robust therapeutic target by using Cas9 and helping alleviate the Huntington's disease. And so I thought this was a rather interesting one for talking about. The next one is a really interesting usage of, and this is a good general example of how we are trying to develop plants to be more robust against bacterial pathogens. And one thing to kind of point out about bacteria, many versions of bacteria in plants, is that they are turning on genes to a really high level to support their own growth. It's like they're telling the plant, make more resources for me, not for you. And then that's what leads to pathology and the propagation of the pathogen. And so one way to try and develop resistance to that is if you can edit those on switches to become off switches that are, driving, what the bacteria are trying to do, you can actually create disease resistance. And so that's what I have here in these diagrams, is showing that, there's this region of a gene where if you target it, it turns off the expression of something known as the me-sweet gene. And what you see in the next picture down is that the edited version, which is on top, is much less susceptible to the Xanthomonas infection. And so this leaf stays more robust. Even the small number of scars that it does develop tend to remain very small and localized. And then the chart at the bottom is showing that compared to, wild type susceptible plants, that these different edited versions have lower amounts of disease in them. And so I think this is a really good example. That's, again, that in and of itself is not novel, but I wanted to point this out because also, how many people have cassava on a regular basis, right? I suspect within this audience, cassava is not like your go-to meal, but especially in Latin America, it's actually a staple part of many diets. It's actually in some cases around 60% of the calories that are taken in, especially in Latin America. And so, I think I want to highlight two things. That one, this is a strategy for developing resistance in plants, but because the CRISPR technology is so kind of easy to use in a sense, even these less commercially, globally important crops do get attention. Well, speaking of banana, okay, so I just have a quick roundup of a few news items that I want to talk about without getting too much into the science. But again, one way to enhance the nutrition of diets is to make hand fruits easier to eat, right? One of the major developments were all these people taking regular sized carrots and trimming them down to becoming baby carrots, right? And so a company called Pairwise is taking this strategy, and they have edited the blackberry to be seedless. I think blackberries are really good, but I don't feel like dealing with the seeds. And that's kind of the same is true for cherries, other really nutritious foods that are fun and tasty, but the fact that they have seeds. And so if you think about watermelon, right? I lived at in my youth, watermelons with seeds was still a thing, and those uh, were tends to uh, not get as much you know usage. Uh, well, again, it has to do with you have breeding populations to make the seeds, and you have to separately maintain your breeding populations, and then you sell the seeds that come out of when they germinate or when they flower. So that's how that works. Another one, there's a company called Tropic Biosciences, and they're really invested in trying to get bananas to, again, bananas, while we love having tons of bananas here, when you think about plantains and other versions of bananas, those are actually a stable crop for many places in Africa as well as in the Asia Pacific ring. And so, that can be like 50% of their calories are bananas. And if these are under a lot of stress from a lot of um, different diseases, especially fungal. And then here's an interesting development is that there's in sustainability efforts and helping farmers make money, there are these things called cover crops. And that is you grow something over the summer, you plant it in the spring, it grows over the summer, you harvest in the fall, and then typically your field lays fallow over the winter. And so there are many, and then one consequence of that, though, is that if you have a lot of rain and have a lot of stuff happening, that can deplete the soil and goes off into river streams and creeks and runs away, and the nutrients go away. And so these cover crops are a great way to maintain soil health, but not many farmers plant them because that's a lot of extra work. But imagine if your cover crop also has productivity that you can sell. And so Covercress is a company that has taken what was considered to be a weed, something known as pennycress, and try to make it through genome editing something that can be sold, grown as a cover crop over the winter. So you plant it, it might grow a little bit, it holds onto your soil, you harvest it in early spring, and then you can sell it for either oil or livestock feed. And so again, this is a great example of this. And this year was the first time that farmers could try and get the seed and trial it out if they were interested in wanting to volunteer. So I think that that's kind of an interesting development for 2024. Again, this is so weird. This is another slide that's not resolving for me. So this target gene, okay, so this is, again, coming back into this larger picture of if we can do editing, if we can change the nature of organisms, are there new organisms that can have new value to say our food sources or even building resources, and that's more sustainable, right? Because it's much easier to grow larger amounts of, say, fungus in a lab as compared to, and in fermenters, as compared to using up a lot of land space like we do in agriculture. And so the example I have here, and this has been around for a little while, this is not entirely novel, but this is a new version of it where they took the fungus Aspergillus oryzae, which we know as an edible fungus. It's something that's present again, for thousands of years that we use for fermentation, protein production. It's been around as a meat alternative. And what they decided to do with CRISPR was to make it more molecularly and engineering friendly. And so that's what I'm showing here on the left-hand side is that, these filamentous fungi, these can be really good for fermentation as an actual food source. But, if you're like me, you look at a fungus and you're like, I don't think that's all that good looking. And the taste of fungi have not evolved to be flavorful. They're not like fruit in that same way. And so what they did was they used CRISPR to really try and make this more amenable to doing all sorts of engineering aspects. So the two that I'm going to just highlight here from the paper, one, ergothioneine is a powerful antioxidant. It's a food supplement that you see in a lot of cases. It does seem to have health benefits. And so they used editing to enhance the expression of a couple genes that are actually native to the organism, but just made them turn on higher. And by turning them on higher, the bulk amount of ergothioneine is just higher on a per weight basis, and so that becomes more valuable. And then what you see on the right-hand side is there actually, so have you guys heard of Impossible Burgers? You guys have heard of that, right? Yeah, so Impossible Burgers, the way those are made is that they found a way to express more hemoglobin in soybean. And then, of course, you harvest the soybean, you extract out the hemoglobin, you might use some of the you probably use some of the soybean itself as a part of the bulk of the Impossible Burger. And because you have this hemoglobin in it, it tastes more like real meat, right? Turns out that hemoglobin is a key part of the taste of meat, as well as, I think to some degree, the texture. And so what they're showing here in this picture is that by enhancing the amount and injecting a heme biosynthesis pathway into these fungus, they can actually just directly cook them and get both the texture and coloration that you want for this meat alternative, right? So as compared to growing a whole field of soybean and putting all this hemoglobin in it and trying to harvest it, you can actually just grow this in bulk and just directly use it into the manufacturing process. So again, this is, I think, a really interesting example of how, this ability to do genome editing just really changes the equation, what we can, or what we want to use as our building blocks for, say, food or other materials. And yeah, Emma Leoch does mention that fungi are trying to disperse themselves by being eaten. I mean, that's a part of their strategy. And so again, we're not talking about some random plant. We're talking about fungi that already have value within the food chain. And this actually comes back to another important idea, is that there are other fungi that are more amenable to genome editing, but because they don't have a what's called history of safe use, in terms of being known to be eaten by people without harmful effects, then they're less likely to get used and get through regulatory processes. And so this particular aspergillus is very regulatory friendly in that case. All right, let's move on to the new tools. And again, this is the one that's even more complicated to talk about than that, but just some takeaways from this. What this group has shown and done, I think, elegantly for one of the first times with CRISPR, has developed what's known as basically a hemostatic protein expression system. Because one of the things that we do with genome editing is we try and say turn a gene on higher or turn it off or have it be expressed lower. And we do this in experimental conditions. But then what happens when you have an organism or or a patient, and then they're undergoing stresses where the expression pattern and how genes are working change all over the place. And that can have big consequences for your edited genes, that they're not able to respond, and they have basically bad side effects in terms of how they are turned on or off under stress. And so what they've developed here is basically the idea of a rheostat. A rheostat is something that when you have a perturbation to the system, it brings it back to where it's supposed to be. And so I think this is really important for that next phase and generation of how we think about complicated expression systems, especially when it comes to certain types of therapies, and most especially when we're talking about engineered yeast and engineered bacteria. That's where it can become really important. All right, so that's an interesting development. And so weird, this slide is not working. Um, let's see, looking at my offline version. Uh, so here is another kind of two interesting things about this. This is combining a known editor and things that can make breaks from before. But what they have that's novel and new is that you remember I mentioned early on that talons and meganucleases, they don't need RNA to tell them where to go. They can be engineered to do that, but they're not very programmable. What this group has shown, and again, the initialization PNA is really important, which what that stands for is a protein nucleic acid. And so it's actually protein, it's actually amino acids, but it actually recognizes the base pairs that complements a particular target DNA. And so what this is doing is creating this like bridge technology where you can actually target these other versions of editors to the right place without needing RNA at all, but making them slightly more programmable. And so that's the upshot here, is that they're more independent. And in fact, for some of the things I'm showing here, I'm going to skip it for now, is that there should be less off-target effects from this type of technology for a couple reasons compared to some of the CRISPR ones. So that's actually a really interesting development. All right. The next new tool comes down to a new way of thinking about editing. And this is where I always kind of, I might be overthinking what to name my talk. But one thing that we do with genome editing is it's not just the genome, it's the epigenome. And so what the epi stands for here is how much genes are turned off versus turned on. And there's a code for this. There's a chemical code for this that's not changing the A's, the G's, the C's, and the T's. It's changing the proteins that are bound onto and are telling genes whether they need to be on or off. And so what this group is showing, what this diagram is kind of complicatedly showing, is that they're using CRISPR in a way to recruit a protein to the on-off switches for genes, and that by recruiting that protein or that fragment of a protein, it recruits other enzymes to turn the genes off. And so that's what you're seeing on the top line, is that you have a gene, it's on, and then as you recruit these other catalytic activated, as you recruit these other enzymes, you turn the gene off. And so they're using this as an example of trying to silence the prion proteins. So prions are prions are proteins that are capable of self-aggregating. And when we think about Parkinson's disease, Alzheimer's disease, and beta amyloid, those are things you might have heard of. Those are considered prion diseases. So if you can make those proteins express less, then you should be able to help alleviate and deal with some of those particular diseases. Although again, one thing I think is kind of notable, and coming back to I just want to do a callback to the microbiome, is there have been a lot of interesting experiments and data recently showing that your microbiome and the bacteria you have there are actually an influence on whether you develop tauopathies, these different brain aggregation diseases. So good news for how we think about and how we might treat and deal with those. Now, the other new tool, the flip side of this, is that this other group did work similar to that, where they're actually turning genes on. So instead of trying to turn a gene off, what they're demonstrating here is that by recruiting a fragment of a protein to promoters, it has this opposite effect of turning genes on. And so I think that's what I'll just leave you with on this pair of slides. The next two slides are doing the same topic, but I needed two slides to, I think, introduce the idea. Uh, in this one, there's a new type of editor known as bridge RNA-guided uh, recombinases. And the takeaway I want you to take away from the diagram is that a combination of the protein, as well as the target DNA, as well as guide RNA, this combination of factors, it complexes and allows to do a specific targeted cleavage event, much like the diagrams I've shown for CRISPR in the beginning. And so, um, and what's, in terms of how they thought of this idea is they just realized that these elements that are in bacteria that are ancient, these are ancient processes where genes try and jump around. And so they're used to guiding themselves and inserting and making breaks in DNA. And so this is just a whole new class of these that are doing it. Now, what this means is this is a completely new tool that's not CRISPR. And so I think one of the important aspects of this is that whatever intellectual property space there is around CRISPR-Cas9 and Cas12 and all these, this ends up becoming an alternative that would have a different licensing regime. Now, the downside is that so far these have only been demonstrated to work in bacteria. They haven't gotten these to work in mammalian cells yet. And that's what I'm showing here on the next slide. Same group, they did a separate paper where they're just showing how they're able to take a target site, they're able to take a payload and deliver it where they want to and make the genetic changes. And then this kind of letter word cloud you see on the right is just an example of how it's just basically making the point that you can kind of target whatever sequence you want. You're not limited to very specific target sequences. So it's programmable, and it's kind of the same way that CRISPR is. So a very exciting development. Very exciting that we have new tools that as people develop one platform, they think more about it and find more examples of it. And then here's an interesting topic that I think may or may not, we'll have to see how much it actually works. But for those of you who are excited about AI, this is also a very exciting development for how to use artificial intelligence by taking all of the known Cas9s and Cas12s and then using an AI to develop a novel one to say, how can we use the information of all these ones that we already know about to actually go back and say, can we make something new and novel with a completely new sequence? And so that's what this company did, and they're showing in the bottom that the process is shown in the first diagram. The diagram on the bottom right is just showing that compared to CRISPR-Cas9, their editors work at editing as well. And so the way this has been promoted is that they're saying this is an open license that anybody can use this, but I think the actual intellectual property aspects of this will still need to be hammered out. But regardless of that, it is a really interesting example of how, if there's any sort of activity that's well characterized, this is an example of how AI can actually help you generate something new. Okay. And then here's another example of, again, I'm kind of a little bit out of thinking about how genes interact with DNA. These are known as hydrolytic endonucleotic ribozymes, or HIREs. So you might have heard of, so the term ribozyme, and when you break that down, it's just saying this is an RNA molecule that is able to do enzymatic stuff. And so we typically think of enzymes as proteins, but there are several examples of RNA being catalytic. And as they went through and looked at these catalytic RNAs that are also, again, these mobile elements that move around and jump around, they actually discovered one that is reasonably programmable, that they can actually do DNA targeting. So this is where the RNA is making breaks in DNA. Now again, so there's good and bad. I mean, there's the really big advantage of this is that they're very compact in terms of delivering them as an RNA. Although to some degree, they're not going to be as robust for, say, adding an editor or adding other stuff onto it. But this is, again, another completely different new class of doing genome editing that's not CRISPR. And we'll see how this one takes off. So I have a couple quick slides to cover the cautionary tales. So as I kind of introduced earlier, interacting with DNA and interacting with cells does have its hazards. And it's really important in some situations that it just not go wrong. Now, in the plant space or in the R&D space, if you have something that goes wrong, you just make sure you have a good way to screen for the right ones and just keep those and use those. But when it comes time to trying to directly edit people's cells or extract stem cells and edit them and then put them back in the patient, you have to be very clear that you're not making deleterious mistakes. So, and yeah, I think Roy is mentioning here too, is that if you just do one infusion and you think, oh, this one infusion will be a way to cure a person, you don't, how sure can you be, or how diligent can you be to make sure that you're suddenly not, you're curing them of one thing, but then helping them develop, say, cancer or other problems later because something goes awry. And so this is a cautionary tale of, again, the key thing here is that they're using a lot more robust analysis tools to see where and how often things have gone wrong. And so these are very complicated experiments. Now, the main thing to take away from the graphics here is that the more red arcs you see, that means the more often something's gone in the wrong spot, or something has recombined in a way to be bad. And on the very far right-hand side, what they're showing is they're trying to make an edit in one spot, and in order to make that edit work more efficiently, they're trying to go back and inhibit DNA repair, right? They're inhibiting NHEJ again. They're inhibiting one repair pathway to make the pathway they want work better. But actually, while they get more targeting, they get more of the change they want, they're also getting more bad consequences by doing that action. And so again, this is, again, talking about every time we think about these technologies and how we combine them, it's important to analyze what's happening. And then this next paper is basically another group that did the same thing. And they had a little more emphasis. Like I mentioned before, when you try and do base editing, one of the advantages of just doing these small little typo changes is that you're not making a double strand break. So people have always assumed you'll have a lot fewer of the bad consequences that are happening all over the genome. And what this group is showing, and the diagram that they have there at the bottom, wherever you see the orange or is examples of where something has not gone quite right and it's aberrant compared to what you're expecting. And so they're showing that, again, in different cell types, different cell lines, different conditions, you can actually have double-strand break type events that you wouldn't expect to even be there. Now, these rates are not super high, so don't get me wrong. It's not something where people are like, oh, this means we have to give up on it. But they do have to find a way to fine-tune it to a safety level. And then this next paper, independent group doing some very similar work. And they just have, I like that, one, I'm just re-emphasizing the same thing that the previous paper did, which is you think you're just doing editing, but you're actually still making double-strand breaks. You're still causing things to go weird all over the genome. But they also had this nice circular diagram, which is just kind of underscores and shows this process where even though they're trying to do some minor change, there are other things going on. And it's kind of a nice flywheel demonstration and diagram of these. So what I will do is stop here to summarize and say that 2024 was another very exciting year in genome editing. We're seeing this bigger and wider pipeline of things in clinical therapy development. There's still an emphasis on those things that are small changes that inactivate a bad version of a gene, but I think we're seeing a little bit more that's going to be more complicated and robust ways to develop therapies. We're definitely seeing more CRISPR foods. We know there's some more in the market. You can actually get CRISPR greens right now from Pairwise that are genome edited mustard that makes a great salad. But we're actually seeing this really interesting convergence of sustainability issues and the ability of how much you can get out of an acre, as well as protect it from pathogens. And that drives a lot of the trait product designs. But then also just how much can we use mushrooms to be food as compared to this typical row crop stuff idea we have? I did not know. So I have I have a quote from Tagline that they're making CRISPR biscuits on British Bake Off, and I missed that one. And again, I think, once people had this understanding of what CRISPR was and where it came from, this very ancient activity of of selfish genetic elements trying to move themselves around, but then became a bacterial immune system. People have gone back to look and see if they can find more examples of this where the IP space, the intellectual property space, and the patents can actually be more unique or have more tools and more opportunities. So I think that's really interesting. And again, we're more and more um, looking at understanding the safety, the causes and consequences of these, and really doing our diligence to make sure that if we're trying to help patients, that we're really creating, again, it's almost always impossible to create zero risk, but to make sure that we have a manageable and as small amount of risk of side effects as possible. And so with that, I will end. I see some questions in local chat, and I'll address those. But right now I'm hitting the hour, and so I'm going to pause here and thank everyone for coming. And I hope you learned something, and I know it's a lot of stuff, but the impression is what we're trying to go for of all the exciting stuff happening in space and how it relates to you. All right, cool. Yeah, good. If someone can take a picture and send it to me, that'd be great and appreciate it as well. All right, so there are a couple interesting questions that are coming through the local chat. Let me go back because I thought I saw one about aging. Oh wait, sorry. Let me do this one from Tuya first, which was, does a gene include its own transport mechanisms? And now again, I think you're asking, does the CRISPR package and machinery that's trying to do editing, it doesn't have its own transport mechanism. And that's why really the whole delivery is this whole topic in of itself, is that you can't just give, it's not like a pharmacological agent where you give them a pill, it disseminates throughout the body. You have to find ways to, because that would just get chewed up. CRISPR and RNA would just get chewed up by the stomach. And so you have to find a way to inject it and make sure it's packaged in a way, because otherwise it would just be a random protein in the bloodstream. So that's where the transport and basically making sure you're mailing it to the right address is a key part of how this technology works. Let's see. I saw a question about whether this applies to aging, and aging, of course, is another one of my key scientific interests where I've done some communications, and aging is very complicated still, but within the CRISPR space, there definitely are some trials having to do with osteoporosis. And there are ways of using CRISPR to understand the aging process, and I think that's where a lot more of the development is. Right now, there's really a hope that now to give you a more technical but exciting answer is that people like David Sinclair and I think a few other groups, they're actually using CRISPR-based tools to actually de-age cells. And you can do that very robustly. There are these things called Yamanaka factors, which was its own Nobel Prize. And you can use CRISPR-type technologies to make a cell younger. But that's not an effective therapy for the whole organism as of yet. But I would say that's one answer for the aging question. Belle, glad you came. Yeah, that's the whole point of Science Circle, to try and get people who are not scientists an appreciation of what's going on in the science world. Let's see. Yeah, okay, so Syzygy, who asked about impact treatments of aging, we're certainly, we're using it more to understand aging, and in some cases we can use it but it's not a practical therapy. Let's see, I think I had a question in an IM. Oh, so trees. I have a question about trees. And so I think it's is the chestnut, the American chestnut. That is one that actually had an interesting history. So I can't go too much into it. So chestnut trees are very susceptible to, I think it's a fungus, it's a blight that came from Asia. And it turns out Asian chestnuts are fairly resistant to it. So they actually have done genome editing to take to take the Asian version of that gene and put it into American chestnuts. But it turns out tree people are extremely conservative and they don't want genome editing to be the source of chestnuts in the wild. And so it's actually a really, really interesting example of it's a solution, but it's not the solution people want. But that has been helpful in trying to do breeding programs that are trying to get those same genes through a normal conventional breeding method. So CRISPR, I could say, was helpful for them understanding that it was feasible to do that. Okay, yeah, thanks for coming, Bill. See you, Mike. Thanks for coming. Maths Club, yeah, keep a reminder to everyone, and actually I will pop this in here real quick too. The Science Circle maintains a web page where we are always advertising and marking when we have events. So keep an eye on that web page. And I'm having an issue here. There we go. So a place to know what's going on all the time. Any other questions? So I think I've gone through the back catalog of questions I saw in local chat. We go a little farther back. Cool. Glad you like the tagline. Yeah, this is a great field. There's a lot of stuff I couldn't cover. Again, I'm trying to pick the things that are kind of novel and unique, but also hopefully relatable. And there's a lot of stuff, a lot of other really interesting things going on. And I just can't cover all the stuff that I've talked about in previous years too. Those you just have to recognize are still ongoing and happening. All right. Feel free to IM me a question. Yeah, I have a feeling next year I'll probably talk a little bit more about the intellectual property landscape of this because of, especially from the stuff we learned from the new tools. Unfortunately, we didn't have any particularly good movies or anything like Guardians of the Galaxies 3 last time that leaned heavily into the idea of genome editing. Oh, Operative Phoenix, you're talking about chocolate. Yeah, so I I hate to be the bearer of bad news, but both cacao as well as coffee, those plants are under a lot of stress. Coming back to the coffee version, I saw a guy from UPenn talk about it a few years ago, and the majority, like 25% of the coffee trees are, or maybe it was chocolate, no, maybe it was the chocolate trees are old, and they're expected to decline in productivity rather dramatically. But it's very hard to establish new new uh, new new trees for cacao, and there's a lot of disease stresses. So I know I know coffee and chocolate are in danger, and so there are people working on it. Um, but was there something specifically you want to talk about? Phoenix. Um, I mean, the one thing that's kind of interesting about the cacao trees is that they actually are amenable to grafting. They're like oranges in that sense. You can actually graft on young branches onto an older tree, and so that we can regain productivity. Yeah, Astor makes a really good point. I don't know. I mean, coffee and chocolate kind of drive it. Um, oh, so here's a question from Bell. The synthetic meat. Um, is synthetic meat beneficial for human health? And what are the potential long-term effects of its consumption on the human body? Well, I mean, let me say that the majority of the meat substitutes that we have right now are plant-based. And so it's just the idea of eating lots of more plants in terms of the effects on human health. Now, the one space where meats are naturally a kind of unique source of certain things are vitamin B12, as well as trace elements and minerals. Or sorry, minerals. And so, you know, the, you'd have to talk to someone who's more of a specialist in terms of nutrition about like completely vegan plant-based diets. But as long as you have milk and some other sources of some of the rare nutrients, you know, these diets are just they're good for you. They're much better than some of the ways in which the meat calories and meat um, things in meat can actually uh, are not as good a diet to have, especially in terms of fat. Right, plants are very low in terms of fat. Now, the other component of that is that the reason why, from a sustainability perspective, you know, you make a cow, and that cow that you eventually butcher and use as much of its meat as you can for human consumption, well, that took how many thousands of pounds of food, of plant that you fed to the cow, right? So the whole idea of it is that by not having as many livestock, you don't have to grow as much food in order to make the meat, that you just eat that plant matter directly, and that way you use less planting acreage, there's less, um, again, methane and carbon dioxide from livestock put into the environment. Here's an oddball little fact about Iowa compared to California. You know, Iowa's population of people, I think, at most a couple million, whereas the population of California is tens of millions of people, right? Well, the state of Iowa actually produces more sewage than the whole state of California. And that's because of all of the livestock that we grow here. And when you think about the consequences of dealing with that sewage, that's a big contributor to the dead zone in the Gulf of Mexico. And so imagine a world, again, let's just take an extreme example of this. Imagine a world in which all of Iowa's land was reclaimed from growing corn and soybean and meal for livestock, then also get rid of the livestock. How much food could we grow that's just directly to the consumer from plant-based food? So, you know, that's a thought experiment. And I'm not necessarily advocating it in that way, but that's an important point to think about. That's how these arguments are made. Yeah, and Tagline does mention that, you know, about 40% of the acreage planted in Iowa actually goes to ethanol production, so it's not even for food. Um, yeah, I mean, there's so many things we can go back and think about sustainability and nitrogen fixation. But yeah, the main thing is, to answer your question, Bell, we use much less resources to feed people plants directly than we do to make plants to feed animals that we then eat. Yeah, no, good question. I mean, I think, and that's where, you know, that's one of the reasons why I gave the example of the fungi, is that those end up being a really nice way to develop plant food sources. And here's a crazy story. If you do a Google search for growing fungi in basement parking lots in France, you'll actually come across a real interesting amount of information where we can actually grow more food with by using what we would call zero arable land in order to enhance the amount of food we make. Yeah, no, I mean, what this is, so, I mean, I'll mention it now because the Farm Bill, the government did not revise and pass a new version of the Farm Bill. And one of the big arguments in there is what foods and what crops do we subsidize? And I think one of the distressing things that we do with our policy is that we don't help subsidize nutritious food for people, like berries, strawberries. There's not food that helps farmers have insurance and robustness when it comes to those nutritious foods. And so I think there's an aspect of the conversation there that, again, if we could, farmers would, I think farmers would happily diversify the crops they make in many cases, but they don't have a business model that's as good as other stuff. Yeah, because the healthy food, again, a lot of foods are hard to grow where you get the economy of scale and have the safety net of good insurance, agriculture insurance policies. So that really ends up being a key part of the issue. So interesting conversation. Write your representative if you have thoughts. Um, okay, so I have a question coming in from IM, and this is kind of an interesting one to talk about cancer. One thing there wasn't anything dramatically new in the world of cancer therapies, but what but something I have covered multiple times in my reviews in the past is this ability of using CRISPR to make immunotherapy for cancer so much more robust. And the way these immunotherapies work is that you take T cells out of a patient and then you program them to attack the cancer. You might use CRISPR to do that. But then you also use CRISPR to make sure that these re-injected T cells stay alive and stay robust in the body for a long time. And so cancer is one of really, again, we're not using CRISPR to target the cancer cells directly. There's still a lot of complicated biology, and chemotherapies have come a long way, and there's still a lot of value for how we can chemically treat the cancer itself. However, in the area of immunotherapies, oh, excuse me, CRISPR has become a really big deal. And so those are known as CAR Ts. Let me put that in local chat. CAR T is the name of those modified cells. So that's an exciting space. I'm sure I'll come back to it because I think, again, when you go back and look at the slide on things that are in clinical trials, I think we had a bunch of CRISPR-based cancer therapies, which really are the CAR Ts. A lot of those are in phase 3. And so I think next year will be the year that we talk about it as being an actual therapy off the shelf, hopefully covered by insurance. All right, sorry. So I missed some local chat here. Yeah, I don't want to get into a big debate about food policy, but I do want to say that, a lot of people are investing in ways to, like pairwise, make nutritious foods more scalable, more growable, something that's good for the pocketbooks of um, farmers that consumers want. And I think that that's a key part of it. But also, even with the stuff that, even if you don't like, you know, certain policies, we're still making all that work a whole lot better. So, there are all these charts out there showing that if we had the yield per acreage of corn that we had in the 1930s, the entirety of the land of the United States is what we would need to plant in order to make as much corn as we are producing now on what is actual like corn farmland. Yeah, well, I think so. Bell talks about, let me step back and say one thing. When we think about developing things for market, you have to realize how fickle consumers are. right? And I don't want to turn this into a big long debate, but people did not eat carrots. People did not eat a lot of carrots. They did not eat a lot of pears until someone said, let's just trim off a bunch of corn material and call it, sorry, a carrot, trim off a bunch of carrot material, call it a baby carrot, and put it in a bag, and then mix that bag with apple slices, right? Pre-sliced apples, as compared to what? Holding an apple and biting into it? I mean, seriously, these are things where people are making money by just kind of packaging them in a different way. And right, I love carrots, but I when I was growing up, I ate carrots. I would even not take off the skin. I kind of enjoyed all that skin flavor, the outer surface of the carrot. And I would eat apples, and I also like pears. And I would just eat a pear. I ate watermelon with seeds in it. And so, yeah, I mean, it's really funny. But these are the considerations that if you're trying to make money in the space, especially in the agriculture space, how are you dealing and responding to what consumers want? And that's a very rational thing. Yeah, carrots are good. Again, beta carotene, they're orange because they have all this carotene in them, so that's certainly very good for the eyes. That's a good example. I don't know about itches. Oh, that's funny. When I was younger, I hated carrot cake, but you put a good frosting on that carrot cake, I'm all in. I am all in. So, Amin asks a question. There are side effects from genetically modified foods. No question mark. And let me give one general answer to that question, which is government regulatory agencies, as well as the companies that are producing those, do not want to have any side effect to anything that consumers are putting in their mouths, or even livestock. I mean, that's not the goal of any of this, of the players who are involved. And so companies, and I can speak a little bit to experience, but I don't want to say too much about the specifics of my company. We do so many things that are both required by regulatory agencies as well as not required, but we go the extra step of asking if we've modified a plant in any way with any sort of genetics, whether it be your prototypical GMO, a transgene, or from a base edit. We do everything possible to minimize any concern about it being not what we wanted to make. And so, the I get there is this theoretical fear of modifying plants and then it does something that you don't want. But the fear of that actually getting onto consumer plates is, I think, to some degree, there's a lot of scare tactics that make you think that's possible. But it's just nobody's desire or purpose. Um, so I think that's kind of my quick general answer. And then I guess the question is, if you actually go back and no, there had been some cases, and I think there was an example where they were trying to do genome editing to increase the vitamin D content of, I want to say rice, but I'm not. No, it was something else where there was an intermediate in the I think it was the vitamin D pathway that got built up in that version of it. But that was easily detectable, and they didn't move that forward as a product, right? So from a theoretical point of view, you can edit something and have something go wrong or go bad. But, you know, but the nature of an edit in and of itself, it's actually what nature does, right? There's nothing in nature that you would say, I should say, obviously at some point plants developed arsenic and are able to make cyanide, right? And so insects don't eat them. Well, as humans, we don't want to eat those either, right? But when it comes to making an edit, the intrinsic nature of just making a DNA change in plants is in and of itself not dangerous for what the plant becomes. I hope that answers your question, Amin. I mean, you know, I gave another talk recently about how there's a long history of plant breeding and plant changes, and that really there's very little to be scared of. And that's even in before regulatory agencies and before all the molecular tools we had to really understand what's happening in cells. Actually, tagline, again, I'm not going to read the whole thing, but just tagline makes a really interesting point that compared to the agriculture system that we had, where, you would crop dust, you would spray whole fields just from the air, pesticides and or herbicides and or fungicides, right? And these are things that we know have consequences if humans ingest them. And so from a genome editing perspective, and even just back to traditional GMO technology, trying to find a way to help plants protect themselves from insects where you never have to spray stuff, that's a win in terms of both sustainability and how much that keeps those chemicals out of the environment that can get into people, animals, fish, streams, water supplies, all these things. And so the major driving force of a lot of things we do with biotechnology in terms of seed products actually helps us alleviate all the things that when you don't talk about something in isolation, there's a net benefit to use less pesticides, to use less herbicides in the environment. So I hope that also answers that question. Oh, so Roy, okay, so Roy asked a question, which is why is it that tasty food is unhealthy and healthy food is not tasty? And yeah, there's some interesting aspects of that from evolution. You're welcome, Amy. Um, you know, sugars were very rare in the environment, and so they ended up but they're a very, very rich source of calories. And so we developed a taste for them, the ability to identify and want them, even before we were thinking, right? This is before we were thinking cerebral type organisms. And then we have a desire for them programmed, but that's because they were rare. They were valuable, but rare. And the same is kind of true for fats. So there are a lot of things that are fats. They're very high in calories. They're very good for us, very tasty. And so now that we have we have through technology and agriculture a plentiful supply of sugar, you know, we still have a driving desire to eat it and taste it, uh, and but that's now in a context in which it's plentiful. Now, I think there are some other things to talk about in terms of, this highly processed food conversation, the additives that go into food that is highly processed. Again, one thing, and I'll just make one mention, this can be a pointer for how to do your own search. Keep in mind that in the United States, the major conglomerate corporations that bought up food companies in the 1970s and 1980s and 1990s, places like Nabisco and um, uh, Kellogg, these major food companies were bought up by whom? Tobacco companies, right? Reynolds, R.J. Reynolds. Again, I'll have to go back and look at all the specific names. But when we think about processed food, there's a lot of stuff that they've learned and scientists have learned that I think has a big consequence. I'll tell you about something I just saw recently, though, and this is to address Roy's question about why. There's some very interesting anecdotal data. And this was there's an interview on NPR's On Point with Magna Chakrabarty, where a guy wrote a book talking about Ozempic. There's an interesting, and again, I don't know how well scientifically quantified it is, but there's definitely observations that people who go on Ozempic start finding highly processed foods disgusting and don't even want to buy them or touch them, and they suddenly develop a desire for fresh produce. And so I think, well, Bella, I don't know. The answer is offhand, I don't know. But I think there's some very interesting questions that come out of that observation, and that these peptides, GLP-1 and GLP-1 peptides. These are these are telling us a lot of interesting things about human biology. Yeah, and actually, to relate it back to the talk too, one thing to keep in mind is that a lot of calories we take in, depending on what sort of microbiome you have, different percentages of those calories can get into your body versus never not get into your body. And so one thing they have shown is that there are certain versions of microbiomes where people tend to eat more calories but actually absorb fewer calories into their body. And so I think that that's another thing that comes back to understanding microbiome interactions. Yeah, sugar is very absorbable, and again, that's the thing too, is that we're trying to sugars are a very fast source of calories, um, and so our body has a way to develop that, but we're also reacting to those fast calories as well, and that's where the whole insulin response system and the appetite system becomes a key part of these conversations. Anyway, I think I've uh, the pace of questions is slowing down. There's a lot more to talk about, but I've also been sitting here for 90 minutes straight. And so I think what I'm going to do is close this out and thank everyone who's still here for coming and hope you enjoyed it. And again, come back next year.